Hello, everybody. Hope you are staying safe. The topic of my presentation is medical cannabis and cannabinoids for impaired sleep, a systematic review and meta-analysis of randomized clinical trials. In terms of disclosure, we did not receive any grant or research support for this work, and the authors declare no conflict of interest. In terms of background and a brief overview, despite limited evidence, medical cannabis is increasingly used for many conditions, with two of the most common being chronic pain and impaired sleep. The prevalence of sleep disorders in the general population is approximately 20%, and roughly 40% of Canadians are suffering from sleep disorders. There are currently two available systematic reviews in the literature. However, they did not conduct meta-analysis to pull effect estimates, and also they did not evaluate the overall certainty of evidence, and one of them has an outdated literature search. Therefore, we decided to conduct a systematic review that addresses these limitations. In terms of method, we did search Medline, MS, Central, and PsycInfo from inspection to January 19, 2021. We included RCTs in any language evaluating the effect of medical cannabis or cannabinoids on sleep, and those that enrolled patients aged 18 or older with impaired sleep, and randomized patients to any form of medical cannabis. Paired reviewers screened titles and abstract, and also did review full text and extracted data independently and in duplicate. In terms of outcome measures, we looked at sleep quality and sleep disturbance as co-primary outcomes, and also other sleep-related outcomes, and all adverse events reported by as equal to 5 or greater than 5 trials. In terms of risk of bias assessments, we use a modified Python risk of bias instrument. We converted all continuous outcome measures to a 10 centimeter visual analogous scale for VAS, with higher score indicating worse sleep. To report pooled estimates, we use continuous outcomes, we used weighted mean differences, and also the modeled risk difference for achieving the MID, which is equal to 1 centimeter for the 10 centimeter VAS. And for binary outcomes, we reported relative risk and risk differences. We also specified a per subgroup hypothesis that larger treatment effects were associated with, for example, shorter versus longer lengths of follow-up, cancer versus non-cancer chronic pain, high THC versus THC and CBD versus high CBD products, and also trials with high versus low risk of bias. We also implemented a newly developed tool called Iceman criteria for assessing the credibility of subgroup effects. We perform a regression for length of follow-up, duration of treatment, and loss to follow-up, and also use grade approach to summarize the certainty of evidence. Here you can see the study flow diagram. As you can see, we identified 2,510 records through searching different databases. And after removing duplicates of 359, we included 2,151. And after excluding the ineligible records, we included 136 for full text screening, and finally we included we excluded 17, which 17 records which were conference abstracts, and also we had 119 articles for full text screening, and finally we included 38 trials for final analysis. In terms of results and general demographic features, 38 publications reporting 39 RCTs with 5,100 patients met eligibility criteria. The median of the average age of the participants was 53 years. Most of them, around 53% of the participants, of the patients were female. The patients with chronic lung cancer pain contained reported in 25 trials, chronic cancer-related pain in 8 trials, Parkinson's disease in 2 trials, and PTSD, sleep apnea, anorexia nervosa, and multiple sclerosis, each was reported by 1 RCT. 1 trial administered inhaled cannabis, and the remaining 38 trial administered oral formulation like drops, capsules, and sprays of cannabinoids. The median follow-up duration was 35 days, and most of the trial were fully or partially funded by industry, 74%. In terms of risk of bias assessment, the proportion of trials that were low risk of bias for each domain was as follows: 82% adequately generated randomization, adequate concealment 92%, blinding of patients, caregivers, and data collectors. All of the RCTs, blinded outcome assessors, 97%, and about 67% of the trials were at low risk of bias for missing outcome data or loss to follow-up, and the threshold was equal and or less than 20%. In terms of the results, for example, for sleep quality, which was our primary outcome, moderate certainty evidence from 16 RCTs with 2,052 patients indicated that compared to placebo, medical cannabis, and cannabinoids, result in a small increase in the proportion of patients experiencing an improvement in sleep quality at or above the MID, which was 1. Model risk difference for achieving the MID was 8%, 95% confidence interval 3 to 12, based on a weighted mean difference of minus 0.43 centimeter on a 10 centimeter VAS. And consistent with these results, for studies that we were not able to pull the data of these trials, all found medical cannabis significantly improved sleep quality compared with placebo. Here we can see the sleep quality on the forest plot for sleep quality on a 10 centimeter visual analogous scale among people living with predominantly chronic pain who receive medical cannabis versus placebo. And you can see the pool estimate here. And in terms of sleep disturbance, use of cannabinoids showed a small increase in the proportion of patients reporting improved sleep disturbance compared to placebo. Again, model risk difference for achieving the MID was 13%. And also we found a significant subgroup effect for chronic non-cancer versus cancer pain. And the test of interaction was significant, P equals 0.03. And high certainty evidence from 10 RCTs of people living with chronic lung cancer pain with 887 patients indicated that compared to placebo, cannabinoids increased the proportion of patients who reported reduced sleep disturbance and modeled risk difference for achieving MID was 19%, based on a weighted mean difference of minus 1.02 centimeter on a 10 centimeter VAS scale. Moderate certainty evidence from 5 RCTs of people living with chronic cancer pain with 1,249 patients also indicated that medical cannabis results in a very small improvement in sleep disturbance versus placebo, and weighted mean difference was minus 0.19 centimeter on a 10 centimeter VAS, and we were not able to calculate the model risk difference for this outcome. Here you can see the forest plot for subgroup analysis of sleep disturbance for cancer versus non-cancer pain. Interaction P was 0.03, and again you can see the results of the pooled estimate here. Minus 0.71, minus 1.04 to minus 0.37. In terms of the adverse events, I just put the selected adverse events. We looked at all the adverse events reported at least in 5 trials, but I just put the five important ones. In terms of nausea, medical cannabis or cannabinoids versus placebo showed increased the risk of nausea and risk difference 5%, 3 to 8, and longer use was associated with greater risk. And test of interaction P was 0.03. And also high certainty evidence from 4 RCTs that followed patients for longer follow-up, more than 3 months, indicated a larger increase in the risk of nausea, risk difference 10%, and confidence interval was 5 to 17 compared to trials with less than 3 months follow-up. And also in terms of dizziness, medical cannabis or cannabinoids versus placebo increased the risk of dizziness and risk difference of 13%, 9 to 20. However, the risk was greater again with longer use, and test of interaction was significant, highly significant, and high certainty evidence from 5 RCTs with 825 patients that followed patients for longer time showed a larger increase in risk of dizziness, risk difference 29%, 16 to 50 compared to trials with less than 3 months follow-up. The results of our study showed that medical cannabis or cannabinoids versus placebo also for diarrhea, high certainty evidence from 12 RCTs with 1,777 patients shows the cannabinoids probably slightly increase the risk of diarrhea, risk difference 2%, 0 to 5. Disturbance in terms of disturbance in attention, as another adverse event, moderate certainty evidence from 7 RCTs indicates that cannabinoids probably slightly increase the risk of disturbance in attention. This difference 2%, 95% confidence intervals 0 to 7%, and vomiting, moderate certainty evidence from 9 RCTs with 1,538 showed that use of medical cannabis or cannabinoids may slightly increase the risk of vomiting, risk difference 2%, 0 to 6%. Here you can see the great evidence profile of medical cannabis and cannabinoids versus placebo, predominantly for patients with chronic pain included in randomized clinical trials. For example, in terms of the smallest study effect or publication bias, we assessed symmetry of the funnel plot and used Eggers test result to assess publication bias when there were at least 10 studies available. For example, we did not rate down for risk of bias when subgroup analysis showed no significant difference in low versus high risk of bias on a component by component basis, or the relative contribution of trials at high risk of bias to pooled estimate was less than 15%. And regarding the precision of continuous outcomes, we rated down when the 95% confidence intervals include half of the MID, and for binary outcomes when confidence intervals include benefit and harms. And here you can see the legend for the table, the gray table. And in this table, you can see the detailed description of the results of 6 studies that were not pooled. Most of them were consistent with the results of the pooled estimate, and only one trial, the results of one trial was not consistent with the results of our pooled estimate. But the result of these trials could not be reliable since most of them are coming from low to very low certainty of evidence. In terms of take-home messages, we found moderate to high certainty evidence that when compared to placebo, use of medical cannabis or cannabinoids results in small improvements in sleep quality among patients living with chronic pain, small improvements in sleep disturbance among patients living with chronic non-cancer pain, very small improvements in sleep disturbance among chronic cancer pain patients, and small increases in several adverse events with a large increase in dizziness with longer treatment. The effects of medical cannabis and cannabinoids on impaired sleep compared to active treatment is uncertain, as evidence is only low to very low certainty. And here are the references. And finally, thanks for your attention. I'm happy to take any questions. Thanks.